Hi, I'm Shirley Tang, a professor of chemistry at the University of Waterloo. It's my great pleasure to be here to share my research activities to develop portable and wearable medical devices through a nanotechnology approach. I'd like to thank Dr. Ip and organizers for this invitation. Lab roots is certainly a new domain for me as a scientist with a background in material chemistry. I'm hoping that my talk will bring some new perspectives to lab roots as well. Our healthcare system is going through a transition from centralized to a more distributed model. Telemedicine, e-health, remote patient monitoring, etc. are at the forefront of this transition. Internet and unprecedented capability to collect, process, store, and sharing data, our patient can access certain healthcare nowadays remotely. Whereas still at the early stage of this transition, a lot of challenges remain. Today, I'm going to highlight just one challenge, which is remote testing. When a patient goes to a hospital, he or she is not only there to see the doctor, they are also there to access trained professionals, central testing labs, to get blood tests done, urine tests done, so that objective evidence can be gathered to assist diagnosis. To have the same quality of healthcare offered through telemedicine, to enable remote patient monitoring, we must have remote testing capabilities. On the left-hand side here are a few parameters that are already accessible nowadays. With the smartwatch, we can record pulse, we can record blood pressure, and for diabetic patients, with a glucose meter at home, they can measure blood glucose level. But for a lot of other diseases, like vascular diseases, the challenge is that the biomarkers are at such low level, remote testing is still beyond reach. So the challenge to medical device researchers is to develop tools to generate test results in outpatient settings without trained professionals, without central testing equipment, but with equivalent diagnostic values to the physician. Now, the research activities in my lab is essentially to integrate intelligent materials and devices into portable and wearable systems for point-of-care testing so that we can address the need for remote testing. At the core of our research are nanocarbon materials, namely carbon nanotubes, graphene. They have exceptional physical and chemical properties. And we develop chemistry for material synthesis, for material functionalization, for forming composites between biopolymers and functional nanocarbon materials to impart biofunctionality, and further incorporate those materials into devices through fabrication processes. And the devices, for example, can be biosensors in array format, and it can be membrane electrochemical sensors, can be incorporated into micro and nanofluidic devices. We've done a lot of proof of concept in the past, and we're doing more system integration to go beyond proof of concept so that prototype systems can be delivered to clinical researchers, that those systems can be tested in the real world. I'm going to focus on project number 1, which is to turn our nano-biosensors into handheld point-of-care blood testing systems for chronic disease patient management, especially for heart failure. Heart failure is highly prevalent globally. 1 in 5 Canadians develop heart failure during his or her lifetime. The number is going to rise 46% by 2030. The rate of hospitalization and the rehospitalization of heart failure patients are extremely high. Traditionally, when a patient experiences symptoms, the patient goes to emergency room where the first blood test can be done, either discharged or hospitalized. And even after treatment in hospitals, the patients often get re-hospitalized within 30 days. Our goal is to develop miniature devices that can enable testing in outpatient settings to break this traditional route so that the patient can receive treatment much earlier, receive diagnosis much earlier, and also use it as a monitoring device to guide management at home. Our system is called Nanobots. It's a system solution, integrates nano-biosensors, microelectronics, and also analytic chemistry method. And showing here is a prototype, a second generation prototype system with an electronic reader about the same size as iPhone 6, or about half, with disposable test strips that can be plugged into the reader and used for fingerprint blood test. The goal is to provide fingerprint blood testing of B-type natriuretic peptide level for early diagnosis and monitoring. And this whole device operates very similarly to glucose meter. There are great similarities in this design. Similar to diabetes, heart failure cannot be cured, so patients have to manage heart failure through a combination of medicine and lifestyle changes. And similar to glucose, blood glucose, we're hoping that BNP can be the biomarker for heart failure diagnosis and management. BNP is an established marker for heart failure with well-understood correlation between the severity of heart failure and the blood BNP levels on the picogram per mole per ml level. Now, the challenge is that unlike glucose, which is high concentration in blood, BNP concentration is 9 orders lower, so that nano-biosensors with exceptionally high sensitivity must be developed to meet this challenge. Now, what is a biosensor? It consists of 3 main components. In our case, the biological recognition element is an antibody that specifically binds to BNP. And the second component is a detector element, or the transducer. In our case, it's a carbon nanotube thin film that translates the binding of antibody and the BNP into electric signals. And the third part is the electronic reader device I just showed you that can read out this electric signal and process and generate a meaningful BNP concentration level. The core technology, the array of nano-biosensors implemented on the test strip, the miniaturization of the electrochemical spectrometer into a handheld device, and algorithms for incorporation of analytic chemical methods and also machine learning. Let's look deeper in the composition or the fabrication process of the nano-biosensors. As I mentioned, the transducer in our nano-biosensor is the carbon nanotube thin film. A challenge to nano-biosensor is the reproducibility. How to produce high-quality nanofilm reliably and using a scale-up process onto a low-cost substrate is highly challenging. And the process we developed will meet this requirement, address those challenges. It starts with silica particles that are loaded with catalysts, which are transitional metals like cobalt and iron. And as you can see that after chemical vapor deposition, which is a process, the nanotubes grew out of them. Each nanotube has a diameter about 1 to 2 nanometers. And those nanotubes growing out of the seeds, which are the catalyst. And we further developed the transfer process that cleans up this nanotube, as shown here, and also allow us to transfer this onto a plastic, a glass, certain type of the low-cost substrates. This is the actual system for the synthesis. This is a low-pressure chemical vapor deposition system. This is not unique to nanotube synthesis. As a matter of fact, this is commonly used in the semiconductor industry. Therefore, the synthesis process we have is readily scale-upable, and it provides uniform in terms of thickness and purity in our nanotube thin film, and is highly reproducible. Here shows the transfer process. On the upper left corner, it's a 6-inch wafer with a carbon nanotube thin film already grown on it. The Raman spectroscopy was used to examine the quality. The peak over here indicate high purity carbon nanotube on the silicon substrate. By the transfer process we have, this thin film can be lifted and then deposited as a whole onto a plastic substrate with patent gold electrodes. And therefore, each thin film, each wafer of thin film can be made into an array of 50 to 200 sensory devices. The principle of operation for the nano-biosensors is that the binding of BMP with antibody on the surface of carbon nanotube modulates the conductivity of the carbon nanotube, and therefore the overall resistance of the nanotube thin film. By recording the electric impedance of the sensory device at various frequencies, a technique called electric impedance spectroscopy, as plotted here, the Nyquist plot of one representative spectrum, we were able to extract the resistance of the CNT film, which is nearly correlated to BMP concentration in the test sample. And these are the atomic force microscopy imaging of the nanotube thin film. Those are the bio-CNTs, clean and smooth. And you can see the decoration of proteins along the tube lines. Those are the antibodies and the BMPs that binding on the surface. And the binding on the surface modulates the conductivity, and therefore more BMP, anti-BMP binding on the surface, the higher overall CNT thin film resistance it will be. Those are test results. On the left-hand side are electro impedance spectrum collected at various time points of the same nanobiosensor. The black squares are collected when the nanotube thin film is clean, and later spectrums are after antibody functionalization, after blocking functionalization, and at certain time points, 0 minutes, 5 minutes, up to 30 minutes, after the exposure of the bionanosensors to a BMP solution. On the right-hand side are plotting of the RCNTs extracted from this spectrum on the left, and you can see that the plotting of RCNT, which is correlated to surface binding of BMP, is an isothermal plot with saturation after 15 minutes, which means within 15 minutes, we can readily, quantitatively detect BMP concentration with RCNT values. Here is the comparison between the performance of our nanobot system to a gold standard immunofluorescence assay. As mentioned, the nanobot system use one antibody as the capture or the recognition element on the carbonaceous surface. The binding of BMP to this antibody translates to the change in RCNT. And in immunofluorescence assay, a pair of antibody must be used. One is the red antibody for capture, and second is the reporter antibody, which is typically labeled with a fluorophore to report the capturing of the target molecule, BMP. And here it shows the correlation between these two methods, which is excellent. The advantages of the nanobot system that by using one single tag-free antibody, the cost is going to be dramatically lower. And the second, it has a much simplified procedure. It's a one-step real-time detection. No wash is needed because the transducer, the nanotubes, only responds to surface binding and disregard the BMP presented in the solution. And in contrast, immunofluorescence assays cannot distinguish between the fluorophores in the solution versus one that bound on the surface, and therefore a thorough washing must be done prior to detection. A third advantage is that by using analytical chemical methods, the system can do self-calibration, unlike the standard immunofluorescence assay that external calibration must be done prior to each quantitative detection. Now here is a comparison between the performance of a nanobot system with a commercial emergency room handheld system. And again, the commercial system requires a pair of antibodies. The reported antibody usually is labeled with an enzyme that can use enzymatic process to amplify signal. And for nanobot, because the nanosensors are so sensitive that no amplification is needed. And also, the nanobot only need one drop of blood that's less than 100 microliters. And in comparison, the commercial emergency room reader needs 250 microliter or more, and therefore venous blood must be drawn. And here shows the correlation between these two detection methods on 28 patient-derived plasma samples. So far, I've focused on one particular system that can specifically detect one biomarker in blood for heart failure patients. Over the past 15 years, though, nano-biosensors have demonstrated the capability to detect a wide range of targets: virus, bacteria, DNA, other protein biomarkers in not only blood and also other body fluids. So I would like to wrap up my presentation by giving you the big picture. Nano-biosensor platforms can potentially offer solutions to a lot of telecommunications for this highly connected world. And finally, I would like to thank my students and my colleagues for their hard work and also the financial support provided by various funding agencies. And thank you for your attention.